Doesn't look like I scared anybody off. That's disappointing. No, I'm just kidding. I'm glad I didn't scare anybody off. I will pass these name things around, so if you could take your name and make sure that you put that up so I know who you are. So today we do have lab. It's going to be mostly like introductory stuff. So we will probably finish early today, but don't count on finishing early like every week. I know it's tight because we only have 10 minutes between lecture and lab, but lab is just upstairs on the 3rd floor, so we don't have to go far. But if you have snacks or a drink, you can always just leave that right outside, and you're more than welcome to, after we do the introduction to the lab, you can always step out and have a snack, step out and have a drink, whatever. I will probably do the same. So, just remove your PPE and you can go out and have your snack. Or you can eat in here too, I don't care. I don't see a sign, so you can eat in here, you can drink in here, whatever you want to do. So we left off on Monday talking about, how can clinicians or clinical lab scientists examine specimens, and we were talking about microscopy, and the idea is that for the most part, what we're talking about in this class is going to be very small. We're talking about bacteria, we're talking about viruses, we're talking about fungi, the majority of which that cause human disease are unicellular yeasts, and we'll talk about some protozoans and other parasites, again, the majority of which are unicellular. And so unless we're talking about things like worms, it's really hard to actually see an agent that causes an infection. So that's why we use a microscope. And I'm sure you remember, or I hope you remember from microbiology, that you can't really see these organisms under the microscope unless you do something to them. Most of the time they're transparent, they're colorless, they're very small, so you may have to stain them. Some of our larger unicellular eukaryotes that can include the yeasts, and yeasts, when we talk about fungi, we'll talk more about how they do this really fun, like they form these mats and these hyphal structures, and our unicellular eukaryotic like protists, you can sometimes see those under the scope without doing much preparation. But other times you might have to add stains or dyes in order to see your cells. We also have, especially for bacterial visualization, some like selective and differential type of stains. So the Gram stain is the classic one. Those of you who took bacteriology here, you know that doing a Gram stain is part of your midterm, like you had to do it, we graded it, right? I found out that I guess Dr. Tao takes the slides and looks at them later. And I should have done that because then that gives you a lot of anxiety. Instead, I just came by and looked right then and told you did a good job. So I'm like, oh man, I'm a sucker. I'm just kidding. I like not having you have anxiety. But it's a differential stain because it allows you to tell the difference between two types of bacterial cells. And in a clinical setting, that can be really important because antibiotics work differently against gram-positive and gram-negative. So it's very important that you know what type of cell you're dealing with so that you can maybe design an appropriate therapy. The Zeal-Nielsen is also called the acid fast. That's one that will allow you to visualize, in terms of pathogens, members of the genus Mycobacterium and the genus Nocardia. It's for those cells that have that waxy mycolic acid outer layer. And then an endospore stain is great for those bacteria that form spores. There are two pathogenic genera, Bacillus. Bacillus anthracis is kind of the one that gets a lot of play, causes anthrax. And Clostridium is the other genus, Clostridium tetani, Clostridium botulinum, Clostridium preferenges, Clostridium difficile. So there are a number of human pathogens where an endospore stain might show you that. You can also do fluorescent staining. Depends on what type of tag or fluorescent tag you have. It can show you different things. Fluorescent staining can also be pretty useful if you have a tissue specimen. Like if a patient has a viral infection, or perhaps you've done a biopsy of a tissue specimen and you're looking for an infectious agent in there, you can fluorescently tag it and it'll show up. So we also can do culture. We don't always do culture. There are a lot of reasons we would not do culture. I would never recommend that you culture anthrax. That will try to kill you, right? It's a biosafety level 3 bacterium. You really wouldn't culture that. You may not want to culture Mycobacterium tuberculosis. It takes like 6 weeks to grow on a plate. You can't culture Treponema pallidum that causes syphilis. It just doesn't grow in culture. Viruses, if you want to culture them, you have to use animal cell culture, so it can be tricky. Fungi are sometimes a little bit slow growing, so you may not want to, or they're very dangerous, biosafety level 3, so you may not. But culture can be a really useful tool for a lot of common bacterial infections. Intracellular bacteria will also be difficult. Some of the major intracellular bacteria we'll talk about will be things like chlamydia and rickettsia. So those bacteria will only grow if they are inside of a host cell, so you'd have to use cell culture. And then viruses will only grow in cell culture. Viruses will not replicate outside of a host. And so when you have organisms like intracellular bacteria, viruses, or very fastidious bacteria, ones that require very specific nutrient conditions, culture can be tricky, if not impossible. But then there are some bacteria like Pseudomonas that's pretty much like, oh, is this water? I'll grow in this. I'll grow on everything. I don't care. So you have kind of that catch-22. So those of you who are interested in going into clinical medicine, again, whether or not you're the clinical lab scientist or the physician, it's important to understand that you need to be able to at least have an idea what infection a patient might have, so you can recommend potential culture conditions. Do you think it's an anaerobic infection? How are you going to sample that? How are you going to transport it to the lab? How is the lab going to grow it? So those are all things that you have to think about. And it's one of those where, sure, you can look up, is this bacterium anaerobic or aerobic, on the Google? But I'll tell you what, if I went into a doctor's office and I was like, I have an infection, what do you think it might be? And the doctor was like, hold on, let me Google that for you. I would be finding a new doctor. So these are things that you kind of have to have accessible to you relatively quickly. And I'm not going to test you on all of these different kinds of media, because when you work in a lab, you will learn about all of these different kinds of media. And we will use some of them in the lab for this course, but we have just very standard media that will grow almost anything, like blood agar. There's chocolate agar, which sounds like it would be awesome, but chocolate is just cooked blood. It's not actually chocolate. You have sabrodextrose agar, which is great for recovery of fungi. And then you have these selective or differential, sometimes both, types of media that will only grow a certain type of bacteria, and it might let you tell different bacteria apart. I know here in bacteriology we use things like mannitol salt agar. It's selective for staphylococcal species, but then ones that can ferment mannitol, like Staph aureus, will be yellow, whereas other staph species like Staph epidermidis will be white. We give you McConkey during your unknown, but I think we use EMB a little bit more. It's selective for gram-negative, and it's differential for lactose fermentation, and you get the E. coli that turn metallic green. So hopefully you remember some of this. And then there are very specialized media that will only grow a certain type of organism. For example, this limb broth will recover Streptococcus agalactiae. And you might ask, well, why are you so interested in only recovering this species of Streptococcus? This is a species that when people are pregnant, it poses a very high risk for a neonate. So you don't really care what other microorganisms they have. right, in the genital tract. You just want to make sure it's not this one. So you test for this one only. It's group B strep. So if you've ever had a kid or you're planning to have a kid, they will test you for group B strep. So Vibrio species, if you suspect whooping cough, right, you can isolate very specific microorganisms in certain situations. So that will come with practice. As you work in the lab, right, in the clinical setting, you will learn more about those types of media and how you can isolate particular microorganisms. So questions on culture. Okay. Okay. You can also diagnose infections through molecular mechanisms. So that would often be something like PCR or derivatives of PCR. Really commonly, what people have been doing in the past few years, right? This is one of the COVID tests, is a PCR-based test. That's not the one you would do at home. It's the one you would have to go to the lab. They'll take your sample, and they will look for the nucleic acids of the virus that says whether or not you have active viral particles within your body. It's a little bit better than some of the other like COVID-based tests because it can tell whether or not you even have just a very low level of the virus inside of the body. The test that you do at home, the rapid test, it can often miss an early infection or if you have a very low level of infection because it's not as sensitive, but PCR is very sensitive. Okay, so if you've ever had a PCR-based test for COVID, that's what they're doing. This is an example or a diagram of how PCR works, but basically the principle of PCR is you have one molecule of DNA and you amplify it over and over and over again. It's a molecular copy machine so that you can detect it. And there are, so before we go to this part, but there are a lot of derivatives of PCR. So you can do a direct PCR on anything that's DNA based. So herpes viruses, human papilloma viruses are DNA based viruses. So you could do direct PCR. But COVID is an RNA virus. And so actually what you're doing when you're doing PCR of an RNA based virus is you first convert the RNA to cDNA and then you amplify the cDNA. So you do a reverse transcriptase PCR or reverse transcription PCR. So there are derivatives of PCR that can be used to determine whether or not you have an infection. If you're interested in doing more of a kind of quick and dirty, right? You're not necessarily trying to be super clean about it, but because you don't want to spend a lot of time, you can also do restriction enzyme digests. So if you have whole DNA, or even if you've done PCR and amplified a particular region of the DNA, what you will sometimes see is that different species or even different strains of the same species can have slightly different sequences in particular regions. And if you digest them with restriction enzymes, you see slightly different patterns. So we call this a RFLP, restriction fragment length polymorphism assay. And so again, this is what it would look like if you ran it out on a gel, and you're looking for similar patterns to tell you whether or not strains or species are related. So looking at this, I would say that organisms 1, 2, and 3 are the same, right? They're probably the same species or the same strain. Organisms 4.
Sorry to interrupt. Did anyone find a pair of keys in here?
Anyone find some keys?
Here.
Oh, bummer. If you don't mind looking around, I mean, you're more welcome to look around.
Sorry about
I don't know where you were sitting.
I was sitting in that row back there.
Okay,
but
yeah, I don't see any up here either.
I don't see any. Okay.
Oh, that sucks.
All right.
Oh,
That's all
I'm
right.
sorry.
Let's see if I can find it somewhere else.
Well, if we
Thank you,
find
guys.
them, we'll put them right up here.
All right, thank you.
Uh-huh. Oh, that's like my nightmare. I'm like, how am I going to get home? I just want to go home. I don't live that far away. I could walk, but I don't want to walk to Denair. I live in Denair. I'm like, walking down Monte Vista, like, please don't hit me. Please don't hit me. Okay. So 1, 2, and 3, right, they have the same digest pattern. So they're the same. 4 and 5 have the same digest pattern. So they're the same. And then 6 is different. So this, kind of looks a little bit low tech, especially for those of you who have taken advanced genetics classes. You've talked about things like DNA sequencing, but it's fast and it's cheap. And this is how scientists can sometimes trace foodborne illness outbreaks. They'll just look for these patterns like, okay, well, all of these people got listeria. We think it's from cantaloupe. Well, where did they get it? Okay, well, we'll test the cantaloupe, we'll test the grocery store, we'll test the delivery trucks, we'll test the fields, and find out where it came from. Real quick and easy to do this. But of course, DNA sequencing, actually getting the order of the base pairs of DNA is going to be very accurate. It's the wave of the future, basically. There are a couple of different types of sequencing we could do. We could do whole genome sequencing. That used to be more expensive. It used to not be realistic. It used to take a long time. But they actually make now portable genome sequencers that you can literally plug into the USB port of your computer and sequence in real time. So if you have a patient specimen, you can sequence it and identify it in real time. And they have been using this, they used it in the Ebola outbreak, 2014, 2016, like through that time period, to diagnose people with Ebola. For bacteria, you can sequence the 16S ribosomal RNA subunit, and that will usually get you down to species and strain identification. So sequencing, as it gets cheaper, as it gets faster, is probably, the wave of the future. But there will still always be a need for, that hands-on laboratory diagnosis. Commonly, to diagnose infection, we can also look at proteins, and we can either look at proteins produced by the microorganism, so we can look for the patient to have microorganism proteins inside of them, or we can look for proteins produced by the host, and those would be antibodies. So you can look for a patient to have antibodies against an infection, or you can look for a patient to have microorganism proteins inside of them. So protein detection can be accomplished in a number of different ways. Mass spec is probably the most complicated, but SDS-PAGE, so running a protein gel instead of a DNA gel. Western blotting, immunoblotting, ELISAs, enzyme-linked immunosorbent assays are very, very common ways to diagnose infection. They can also allow you, especially if you're looking at patient antibodies, you can follow the course of the infection. Are they early in an infection? Are they late in an infection? How is that infection proceeding in the host? And as we talk more about each individual pathogen or each type of infection, we'll talk more about the way that you would diagnose that one. But those rapid tests, let's go back to COVID. Those rapid COVID tests that you have are protein-based tests. And what they have inside of them are antibodies against the COVID-19 viral particle. And so what you're doing when you swab your nose is you're hoping to grab any viral particles that are inside your nose, right? You swab, you touch your brain, like it's very uncomfortable. Mix it with solution, put it on the thing, and it runs through. And what happens is if you have COVID, right? If you have the viral particles in your nasal secretions, they will interact with antibodies on that rapid test, and that's why you see the color change. If it doesn't detect any, like let's say you swabbed and it couldn't find any, that's why you don't get a line. It doesn't mean you're negative, it just means you don't have enough to be detectable. But when it turns positive, it's positive, okay, and you've got the virus. Pregnancy tests work the same way, rapid flu tests work the same way. They're looking for proteins that your body has produced, and they'll tag with an antibody. So again, these are some of the different immunological assays that can be done. Again, really commonly in clinical settings, ELISAs and Western blots, because they're pretty fast. They're pretty easy, relatively sensitive, don't require a ton of training to be able to do them, and don't require really expensive equipment or reagents. But as we talk more about, again, the different infections, we'll talk more about how they're diagnosed. They do give you some examples here for ELISA and Western blot, things like rotavirus, HIV, Lyme disease. A lot of those are diagnosed that way. And so antibodies, again, are important. Those of you who have taken immunology, you already know. Those of you who have taken bacteriology, you've talked a little bit about antibodies, but they are specific for various antigens. And there are two different types of antibodies. There's monoclonal and polyclonal. So monoclonal are ones that are made in the lab. This is not a natural phenomenon. They are very, very specific for one specific epitope. It would basically be the equivalent of if you have, let's say, COVID. It's one portion of one protein and all the antibodies are the same for that one portion of that one protein. It's not super helpful unless you're using it for clinical purposes. So monoclonal antibodies are actually very commonly used in therapies. So if you're watching TV and they're advertising a drug and it ends in MAB, M-A-B, it's a monoclonal antibody. So it's an antibody-based therapy. And they can be used to treat things like rheumatoid arthritis, for example. So they're made in the lab. But what our body makes are polyclonal. Our body says, I don't recognize this. I'm going to chop it up into a million little pieces. And your body will make antibodies against all of those 1 million pieces. And that's much better because you get a really robust response. Cool, I see piece A over here, piece B over here, piece C over here. You get a really nice response. Now the challenge can be, and again we'll talk about this when we talk more about specific infections, sometimes, not always, but sometimes pathogens have antigens that look a lot like our proteins. And what can happen is our body can make an immune response to those proteins that look very similar to our proteins. And so you can have a situation where a patient's antibodies will now attack their own cells. So we do sometimes have patients who have an infection, clear the infection, and still experience some symptoms of an autoimmune condition. So there are some infections, especially like Campylobacter, some of the staph infections that are associated with the development of autoimmune conditions. Again, not every patient, maybe not even most of the patients, but some of the patients can develop autoimmune complications after infection. Again, I'm not going to ask you about all these details. We'll talk more about them as we talk about the infections, but we're taking advantage of the principle that antibodies and antigens interact. We can do that in tissue samples. So what you have here in that panel is localization of herpes simplex virus in nerve cell tissue. So herpes, especially in neonates and infants, can cause encephalitis, can be fatal because it will go to the brain. And so each of those spots that's lit up is where the herpes virus is inside of that patient's brain tissue. Flow cytometry is another type of immune assay. It actually separates cells out. So you can separate cells out by size, and you use antibodies to pull them out. One application of this is how you can count a patient's CD4 positive T cells when they have HIV. Because the clinical progression of HIV to AIDS, part of it is dependent on the concentration of T cells in that patient. Once the concentration of CD4 positive T cells dips below a certain number, combined with a couple other factors, we say that that patient has now developed AIDS instead of HIV. And so you can separate cells out based on immune markers, size, et cetera, using this machine. So this could allow you to pull out, all the cells that the patient has and tell how many are CD4 positive T cells. ELISAs are super common. We'll talk more about how they work when we get into the ELISA section. And the same thing with Western blots, super common. And we'll talk more about how they work when we talk about the infections that are diagnosed that way. So this again is just a list of some of the serological assays. I'm just trying to introduce you to these concepts so that when they come up again over and over throughout the term, you get more and more familiar with them. I'm not expecting you to remember the protocols of how to do all of these things because we're not doing all of these things. But just kind of understanding the basic principle of serology versus nucleic acid versus culture versus microscopy, pros and cons of those techniques, and when you might use one versus the other. So serology can be useful to ID infection, evaluate course. So one of the things that we see in patients is when they have first developed an infection, it can actually take 7 to 10 days for antibodies to appear. So let's use an example of HIV. If you know that you have been exposed to HIV, let's say you know you were exposed yesterday and you go to the hospital today and you want your blood tested, they can't help you because you won't have had enough time to develop antibodies. 7 to 10 days is when the first antibody response will happen. And generally what we see is the first antibody that's found in high concentration is IgM. Okay. After a period of days, weeks, the IgM starts to go down and the IgG comes up. And so if you see a patient that has way more IgM than IgG, they're early in the infection. More IgG than IgM, later in the infection. If a patient has just gangbusters amounts of antibodies, it's probably a secondary infection. It's not the first time they've seen it. So they know how to respond, right? If you get infected with the same thing over and over again, your body responds better every time. It responds faster and it responds stronger. And so when you're reporting antibody concentration, you call that a titer. And I imagine that most of you will probably never have a titer done. There are some situations, weirdly enough, I've had 2 titers done in my whole life. One, when I started working for the Army, they were like, have you been vaccinated for MMR, measles, mumps, and rubella? I said, yes. Do you have proof of that? I'm like, no. So they took my blood, and they were more concerned about measles. They looked for antibodies against measles. Do I have that? And I did. I had a high enough titer, a high enough concentration that they didn't force me to get vaccinated again. And then during pregnancy, one of the things they look for is, do you still have antibodies against rubella? So the R part of MMR. Rubella can cause congenital deafness and blindness in the fetus. It can even cause spontaneous abortion or miscarriage. And they were like, you actually don't have any antibodies to rubella. And I'm like, oh, okay. So I did have to get the vaccine after all. Should have just got it when I worked for the Army. But that's what you're looking for with titer. Does a patient have antibodies? Do they have a sufficient level of antibodies? Do they have a sufficient titer to respond to an infection? When the patient begins to produce antibodies for the first time, we call that seroconversion. Basically, their serum now has antibodies against that infection. And then the secondary response is anamnestic. It's just a secondary response. Your immune system remembers that you've been infected and responds. We most often use serology to diagnose viral infections because viruses, you can't really culture them, right, unless you're proficient in eukaryotic cell culture. I'm not. Hate it. My stuff always gets contaminated with bacteria. I say it's on purpose because I love bacteria. It's not on purpose. Okay, so you have to be proficient in eukaryotic cell culture. It's challenging. Right? Many viruses don't have any treatments, right? Don't have any preventatives. Not safe to work with in the lab. Viruses also too small to see under the microscope. So how are you going to diagnose them? Well, you look for the patient response. That's very, very common. So Epstein-Barr, this is mono. We'll talk about all these, they're fun. So it's one of the herpes viruses, rubella, the one I already told you about, that can cause congenital issues. In adults who get rubella, not a problem. It's really pregnant women are who we most worry about. Hepatitis A, B, C, D, and E, HIV, human T-cell leukemia, which is another retrovirus, and then arboviruses, so things that are transmitted by mosquitoes like West Nile and Zika. That's how they diagnose them. Okay, questions? It's a lot more fun once we start talking about all the different pathogens. I really like this class. Questions about serology, basics on diagnosing infections. You have a lot of tools available to you when you're considering diagnostics. And so that's why you as the clinician have to be really cognizant of what you think a potential infection is so that you can make the best choice for diagnosing that infection. Like it'd be kind of silly to look for antibodies against Staph aureus because everybody has Staph aureus, like everybody. We'll do it in the lab. You're gonna swab your own self, and I pretty much guarantee that probably half the class or more will find Staph aureus in your nose. Whether or not it's MRSA, that's the challenge. Mine is. I was like, man, I worked with Staph aureus for a really long time, so I'm like, well, it's not a surprise. Okay. But, something like Zika is not something that most of us are exposed to very frequently, so if you have antibodies against Zika, that's kind of telling. Yes?
How exactly do they find concentration of the antibody in these titers? Like what method do they use?
It depends. It can be an ELISA. An ELISA can be quantitative, so they might do something like that. A Western blot is more of a semi-quantitative, but for a Western blot, if you do, if you have standards, like a known control, you can sometimes do it that way. ELISA is usually the best. They'll often do fluorescence or some sort of color change, and you can actually measure that using a machine to measure the color change, and then you'll have a standard curve where you would report it that way. I mean, 0 is easy. It doesn't have it. But when you start actually talking about the different concentrations, yeah, that's a really good question. But they will have some sort of standard, either colorimetric assay, fluorescent assay, something like that. Okay. So again, anytime you have questions, please feel free to ask them. You don't have to wait. So just kind of, a little bit of overview of the immune system in general. This class is fun for me because I really like microorganisms. This is how we fight microorganisms, but we get to spend the rest of the class talking about how microorganisms are trying to kill us, which is, in my opinion, a little bit more interesting. I've taught immunology before, but what the microbes are doing, I think, is more fun. But we're all alive today, so that means that at least at some point our immune system has had to do something to protect us. And what our immune system does is it basically has 2 different branches. They do interact. They're often dependent on one another, but because we're people and we like to categorize things. We categorize our immune system into these two different branches. We have our innate immune response, and we have our adaptive or antigen-specific immune response. Our innate immune response is what we were born with. It does not care what infectious agent is coming in. It just wants to protect you. It is not specific for staph or strep or the flu or whatever. It is just trying to protect you. And it's sometimes called our first line of defense. It's what a microorganism will encounter when it first encounters us. And let's say, for example, or I guess for the purposes of this class, the microorganism got past our innate defense. It breached the skin. It got in through the mucous membrane. It wasn't destroyed by your tears or your saliva. It wasn't brought out by your mucociliary escalator. A microorganism has gotten into your body and it has established an infection. Well, what happens next is your adaptive or your antigen-specific immune response. And your innate response will still be working. It's still trying to help clean up. It's still trying to help recruit more cells to reach that area. But your adaptive immune system will also kick in. And what it's trying to do is figure out what this is and design a specific targeted attack against it. This will be where you have B cells that will eventually differentiate to plasma cells and produce tons of antibody. You have T cells, numerous different types. Some of my favorites include cytotoxic T cells, which kill other cells. And all of the molecules that are produced by those cells. The adaptive response takes longer, okay, because it is so specific. But the adaptive response also remembers. Oh, we've seen this before, right? We know how to fight this. Your innate immune system is like, whatever, I'm gonna fight. Let's go, let's fight. Your adaptive remembered. When we're talking about the immune system, yes, we do talk a lot about cells, macrophages, right, as part of the innate immune system, dendritic cells, those phagocytic cells that just go around eating everything. We talk about B and T cells as part of the adaptive immune system. But we also have a lot of molecules that are produced by these cells and others that are secreted into the body when there's a problem. And so, of course, in this class, we're talking about a problem in the context of infection. But a lot of, especially the innate immune response, is very similar to what happens in an injury. So the innate immune system responds to injury and infection. And so we have molecules called cytokines. These are kind of the major soluble molecules of the immune system. They will be produced by cells, travel through the bloodstream, and cause something to happen. What that something is depends on the cytokine. So we have a specific type of cytokine called an interferon. Interferons are often produced in response to viral infection. They're part of the body's just general, non-specific antiviral response. We also have chemokines as a branch of cytokines. Chemokines attract other cells to the area. So let's say you've gotten a cut, it's got an infection, it's probably staph, because let's be real, that's pretty much what all cuts get infected with. The cells in that area are going to release chemokines to recruit more cells. Hey, there's a problem here. We need more cells to come help clean up this infection. Sometimes people will call cytokines like pro-inflammatory cytokines. That generally means that you're going to experience symptoms of pain, redness, swelling, and heat. It can be a localized heat, but cytokines are also responsible for turning on body heat. They raise your body temperature. Well, they tell your hypothalamus to raise your body temperature. Cause a fever. So these are really important molecules to help us fight infection. What they're showing you here, again, those of you who have not taken a class in immunology, this might be like, what the heck? If you've taken immunology, maybe it is still what the heck, but hopefully not. Hopefully you're like, oh yes, I remember this. All of our immune cells come from the bone marrow. So we have a hematopoietic stem cell. Well, hopefully more than one. You have hematopoietic stem cells in your bone marrow. They are self-renewing, and they will eventually give what are called myeloid progenitors and lymphoid progenitors. Very, very broadly, we most often say that cells of the innate immune system come from myeloid lineage. Cells of the adaptive immune system come from the lymphoid lineage, except for natural killer cells, but that's okay. So this is how you get red blood cells, platelets, your different phagocytic cells, okay, and then your B and T cells. So this process of being able to do this is critically important. So people who have, there's like two different kinds of leukemia, AML, acute myeloid leukemia, ALL, acute lymphoid or lymphoblastic leukemia. Do you have cancer here or here? And that devastates their immune systems. People who do bone marrow transplants, right? They wipe out, they kill all of the cells in the immune system to hopefully give you some good ones. Here you might notice it says thymus. T cells will leave the bone marrow, like immature T cells will leave the bone marrow, and they eventually will mature in the thymus. That's why they're called T cells. B cells, they mature in the bone marrow. That's not why they're called B cells. They're called B cells because they mature, they were first identified in birds, and they mature in an organ called the bursa of Fabricus in birds. Just happens to also be the bone marrow in people, so that worked out. But you have normal counts, and so this is another thing that as a clinician you might be looking for, the normal counts of white blood cells. Elevated counts of white blood cells can often signal that a patient has an infection. Their body is producing more white blood cells. Why? Probably because they have an infection. But you can look at the different kinds of cells. You learn to recognize these under a microscope. What we'll do in the lab at some point is, I don't have human samples, but I have bird samples. I like birds. And you can identify, right, their red blood cells versus their different kinds of white blood cells. We're going to do it in the parasite section because I have samples of birds that are infected with blood parasites. So it's kind of fun to look at. Only difference is in birds, the red blood cells have nuclei. In humans, we don't have nuclei. So you will see something very similar if you're looking at human blood smears. You see those red blood cells and then all the white blood cells. So identifying that a patient has an elevated white blood cell count can be indicative of an infection. A skewed ratio of cells can be indicative of some kind of problem. Too low of a white blood cell count can be indicative of a problem. So counting the white blood cells in a patient can be really important. We'll finish off talking about just kind of broadly some of the organs of the immune system, and then we'll pick up on Friday with more. But we have our primary organs, basically the primary organs are where the lymphocytes mature. So lymphocytes is B and T cells, leukocytes is just white blood cells in general. So in the bone marrow, everybody's produced in the bone marrow, so the bone marrow is really important. But the thymus is where T cells mature. And, you know, again, part of that maturation process involves, will you bind to something? So B and T cells have to prove that they will bind to something. If they can't bind at all, they're removed. Will you bind to us? Do you bind to yourself? Those should also be removed because that's an autoimmune condition. So if a B or T cell proves that it can interact with something but not the host, then it's allowed to leave and go out into circulation. If everything goes well. Secondary lymphoid tissues, a lot of what occurs in secondary lymphoid tissues is what we call antigen presentation. Those cells of the immune system that have done phagocytosis, virally infected cells, tumor cells, they all will show antigens on the surface. And cells that are patrolling around, B and T cells, can encounter those antigens and begin to mount an immune response. Commonly that occurs in the lymph nodes. So there are a lot of infections that will lead to swollen lymph nodes. Plague does it because you have a lot of the cells rushing to the lymph nodes. They know there's an infection. Right? Let's go to the lymph nodes and start presenting antigens. Mono or Epstein-Barr will cause swollen lymph nodes up here because it's a respiratory tract infection, so you'll get them in the throat. Spleen, great for filtration. You have lymph nodes all over your body. You have lymphoid tissue in the lungs, bronchus-associated lymphoid tissue. So you have cells that are patrolling there. You have in the gut, they call it the GALT, gut-associated lymphoid tissue. You have urogenital lymphoid tissue. Your whole body, right, is trying to protect itself from being infected. So you have a lot of areas of your body that could theoretically be considered to be secondary lymphoid organs and tissues. So again, the immune organs, this is from the older edition of the textbook. The newer edition of the textbook sometimes reformatted the tables in such a way that it was very difficult to put them in the slides. But the content of the table in the old and the new is the same. So if you have the older edition, you'll recognize this. If you have the newer edition, it's the same. The thymus is required at birth for T cell development. It's the site of T cell maturation and development of central tolerance. That means it learns to tolerate you. Bone marrow, source of stem cells, B cell maturation and development of central tolerance. Lymphoid, so the reason you might sometimes get swollen lymph nodes, it's where B cells can proliferate. So B cells can replicate. They can turn into plasma cells and start secreting antibody, and they can also become memory cells in there. And then there's also a T cell zone within the lymph nodes. In the spleen, you do have another area where you can get B cell development. You also have a T cell zone, and then you have filtration of the blood. When people have spleen injuries, that can be really dangerous. They can bleed out internally because there's so much blood flowing through the spleen. But a spleen is helpful, not necessary. So people do live without their spleen if it's been removed because of illness or injury. And then you have all of your respiratory tract, your urogenital tract, your gastrointestinal tract, anything that's a mucosal surface broadly. They call it the mucosal associated lymphoid tissue, the MALT, but you can separate that to the gut, GALT, the bronchus, BALT. Whatever kind of acronym you want to use is fine. And the skin actually serves as an immune organ because you have, one, that barrier to everything. You have the production of antimicrobial peptides, but you also have immune cells that are hanging out in there, waiting to see what's happening and see if they need to act to protect you. So when we pick up on Friday, we'll talk a little bit more about the lymph nodes, the spleen, the MALT, GALT, and then we'll also talk about vaccination and a little bit more detail of innate and adaptive immune system. Questions, comments, concerns? So again, I'm not expecting you to know the immune system in as much detail as you would for immunology. That's totally fine. And we'll talk more about what parts of the immune system respond to each individual infection as we go. So yes, I will see you all in a few minutes. We're upstairs in 331. Oh, you can bring